Hello everybody, I'm Tapa. I'm a postdoc at Princeton and today I'll tell you about bacterial motility in porous media. This work has been done at Professor Sujit Datta's lab with Daniel and Jenna both focusing on modeling bacterial chemotaxis in porous media. Bacteria are nothing new. We know about their existence for past 4 centuries and we study them mainly in homogeneous material. But in reality, bacteria live inside our intertissue pores, in our gut mucus, in soil and sediments. These are all complex porous environment. So if we want to understand how they behave in such environment, the first thing we need to do is to understand how they move in such environment. And to do so, we have developed a transparent porous media by simply taking these transparent hydrogel particles, swelling them in bacterial cell growth media, and packing them really close to each other. So bacteria can move in the interhydrogel pore space, which we can control by simply changing the overall polymer fraction of the porous media. When we watch how cells move in this porous media, we are completely blown away because the cells do not move by a run and tumble motility. Instead, these cells get trapped into this tight pore space. They constantly reorient their body and eventually find a directed pathway where they can hop to the next trap. So they are hopping between the traps. So what sets this hopping and trapping? Turns out the hopping is set by pore space geometry, while trapping is set by a competition between pore scale confinement and cellular activity. We can separately measure these hops and traps, which gives us a revised view of motility where the long-term diffusivity should be given by the hop length square over trap time. We verify this hypothesis by injecting a bolus of bacteria inside the porous media and measured the macroscopic diffusivity. Our measured diffusivity matches with our predicted diffusivity within a factor of 3. So by simply looking into single cell motility, we can predict the long-term diffusivity of bacteria in porous media. But bacteria live as a population inside porous media. How would you possibly create a dense population of bacteria with design, size, and shape embedded inside porous media? Fortunately for us, our porous media do have some self-healing material properties, which allows us to 3D print bacterial structure deep inside this porous media. To start with, we create a cylinder of bacteria which is embedded inside this porous media, and you are looking from the top down, where each individual pixel represents one single bacterium, and this overall field of view is about thousands of cell lengths. So when you watch this over time, we found these bacteria form these beautiful traveling waves where thousands of cells collectively move over thousands of cell lengths. So what's happening here? If you zoom in, you will see that these traveling waves are not due to growth. Each individual cell is moving, but the traveling front moves much, much slower compared to these individual cells. And if you look from the side, you will see that once we create the cylinder of bacteria, they expand as a hollow cylindrical shell. So what's happening here is once we create the cylinder, cells from the periphery start to eat their nutrients around them, eventually create a gradient of nutrients here, and they start to follow their own nutrient gradient. They chemotax towards their own nutrient gradient. We have found that this traditional mechanism of chemotaxis in liquid culture, where cells can simply elongate their runs towards a more nutrient-rich zone, doesn't hold in porous media. Cells cannot hop longer because hops are set by pore scale geometry. But the secondary mechanism of chemotaxis in liquid culture, where cells bias their reorientation amplitude towards the more nutrient-rich zone, is the dominant mechanism of chemotaxis in porous media. And with that, I would like to say thank you, and I would like to take any questions that you might have.